Good morning. My name is Saraswati, and this is my final capstone presentation for the Monell Science Apprenticeship Program entitled Computational Genomic Analysis of Evolution in Taste Receptor Genes in Carnivora and Cetacea. In this talk, I will be discussing the following main points: the evolution of taste receptors in carnivores and cetaceans over time in accordance with ecological changes, major genetic changes in the coding sequences for a group of genes which were OTOP1 to 3, POU2F3, TRPM5, and GUSTDUCIN. I will then talk about the evolution of these taste receptors in accordance with selective pressure present in the environment. Here I discuss these receptors in further detail. OTOP1 to 3 are membrane proteins that play a sour taste and the cellular transduction of sour taste. POU2F3 is in charge of taste bud differentiation during embryonic development. TRPM5 is a receptor involved in taste transduction of all types of taste. And GUSTDUCIN, also known as GNAT3, is a G protein involved in the reception of sweet, savory, and umami tastes. Prior research has shown that many carnivores and cetaceans have lost their ability to perceive sweet taste in foods, and that some, which swallow their food whole, may have also lost their perception of bitter and savory tastes. Thus, the presence of particular taste receptors may depend on the feeding strategy of the species in question and the type of food consumed. The process by which functional genes become obsolete by function of selective pressure and the accumulation of mutations, as shown in the picture in the slide, is known as pseudogenization. This process may lead to the formation of vestigial structures, which are either residual or entire structures present in the body of an organism that have no given function because they're no longer needed. In other cases, these structures may disappear altogether. The objectives of this investigation was to determine if selection pressure had been relaxed in any cetacea or carnivora taxa with regard to taste, identify mutations and pseudogenization in key taste receptors in cetaceans and carnivores as determined by intact and inactivated genes, and better understand from a bioinformatics perspective the evolution of taste receptors with a focus on marine mammals. First, it was hypothesized that both pseudogenization and mutations will be exhibited in the genomes of Cetacea and Carnivora with a focus on the OTOP1, OTOP2, OTOP3, TRPM5, POU2F3, and GNAT genes. It was also hypothesized that the genes will be completely pseudogenized for the most part in Cetaceans, while partly or not as much pseudogenized and mutated in Carnivores. 10 species from the orders Carnivora and Cetacea were selected for each gene. Then, the mRNA sequences corresponding to the particular genes and genomic sequences pertaining to the species were obtained from the NCBI database. These sequences were then analyzed using MegaEts, a statistical software package with the objective of conducting phylogenetic and evolutionary analyses. These sequences were aligned using this software, and mutations and discrepancies were identified and marked. Afterwards, pseudogenes were identified and several statistical analyses to draw conclusions about the evolution of these genes were identified. In the analysis, Mus musculus, the common lab mouse, was used to compare the species studied to an external reference. Here is the phylogenetic tree for OTOP1, a transmembrane protein involved in the perception of sour taste. This tree shows the relatedness of the DNA sequences of the species shown. In the upcoming trees, pay attention to species names that will be circled. This will indicate if those species exhibited pseudogenization. This particular tree indicates that Panthera tigris is the outgroup. It can also be seen that the carnivores and the cetaceans are predominantly differentiated into separate clades. All of these species have a common ancestor, and this chart corroborates prior research showing that OTOP1 is less functional in cetaceans than it is in carnivores, although not completely pseudogenized, as you can see because of the significant divergence between the two orders. Here we have the phylogenetic tree for the OTOP2 gene, and a similar trend can be seen here. However, here the difference between either order is clear and Canis lupus is the outgroup. Pseudogenization did not occur for these genes either according to the alignment analyses that were conducted in MegaETS. For OTOP3, pseudogenization did not occur here either and no outgroups were found. However, there is less of a distinction here than in previous trees between the two orders, which are Cetacea and Carnivora, as mentioned before, and this indicates that there was less of a selection pressure for species in the two orders studied to lose the function of this particular membrane protein, meaning that it's not obsolete, that it's in fact essential, and its presence in most or all of the species of the order studied. For this gene, GUSTDUCIN, also known as GNA3, an outgroup was found for Orcinus orca, which is the humpback whale, where pseudogenization did occur. Here, it was evident that the majority of land-borne carnivores had vastly similar gene sequences with regards to this particular receptor. Many seaborne cetaceans lost functionality in this receptor, and for this reason there was little to no similarity between many cetaceans and carnivores. There also appeared to be a divergence between the group of carnivores closer to the cat, like puma and acinonyts, and those closer to the dog, Canis lupus. So here you can see that there was a significant level of pseudogenization in the genome, in the GUSTDUCIN gene of Canis lupus familiaris and Calorinus ursinus. TRPM5 was also found to be extensively pseudogenized. The exact type of pseudogenization will be discussed in the upcoming slides. Here, there was a significant level of divergence between the carnivores and the cetaceans. Once again, the humpback whale, or Orcinus orca, was an outgroup, indicating that it had undergone the most changes with respect to other species. TRPM5 was significantly pseudogenized in many species pertaining to the group of seaborne cetaceans, indicating that these species had little to no need for functional taste transduction. And these species, as you can see here, were Neophokina aegiorentalis, Delphinopterus lucus, Balinoptera acutostrata scamoni, and Orcinus orca. POU2F3 shows similar pseudogenization and divergence between cetaceans and carnivores. In this case, Mus musculus is the outgroup having the lowest commonality with all the other species. The species that exhibited significant levels of pseudogenization in this gene were Orcinus orca, Monodon monoceros, and Delphinopterus lucus. I would like to acknowledge my mentor, Dr. Peihua Zhang, and Dr. Joel Mainland and Dr. Paul Breslin, as well as the entire staff of Monell for their help and this opportunity.